In this video, we will discuss about how this endosomes helps in the proteolysis of the ECM or the extracellular matrix. So before we go into the topic, let us first understand what does this invadosomes means. So the invadosomes is the combination of two processes, that is podosomes and invadopodia, and both of them are actin-rich processes. And as we have already discussed this Invadopodia in our couple of our previous videos, in which we have covered this Invadopodia from the point of view of cancerous cells migration, how this Invadopodia helps in the cancer cells towards migrations. In this video, we will cover another process similar to Invatopodia, that is Podosomes, and what are the difference between Podosomes and Invatopodia, and some of the internal mechanisms. So let us start with this Podosomes. The Podosomes also its function is to degrade the extracellular matrix, and how it degrades the extracellular matrix, some of the processes are shown here. That is, it will form a F-actin, and along with this along with the cap, that is plaque protein, and these combinations will help in the degradation of the extracellular matrix nearby the cells or nearby the plasma membranes. And the other combinations that is formed is by attaching this DNAs to this plaque proteins. And how this DNAs is attached, the motor proteins is not yet been identified. However, they are transferred from centrosomes through microtubules, and which motor protein helps is not yet been cleared. So, and these combinations, the function and the efficiency of these combinations is further better than these preliminary conditions, because it has DNAs in it. So DNAs will enhance the ECM degradation's activity, as it will cleave off the DNA of the cellular matrix. And the further upgradation of this stage is that the matrix metalloprotease protein, MT1 matrix metalloprotease protein, will be transferred or to this complex to form a more enhanced complex, and this transfer is done with the help of motor protein, that is kinesin 1. And thereafter, this is the more advanced complex formations that can help in that has a higher efficiency to degrade cellular matrix. And the last step is that matrix metalloprotease protein MT1 can also degrade the cellular matrix on its own, and this MT1 MMP is transferred by the motor protein dyline. Dyline also helps in transport of matrix metalloprotease protein with the help of RAB5A on its own, and some people also thinks that dyline also helps in the transport of matrix metalloprotease protein MT1 with the help of RAB14 and RAB22 to form this complex. However, there is no general consensus has been formed about this motor protein, whether it is dyline or something else. This is about podosomes. Now let us understand some of the internal mechanism of Invadopodia, which we have not covered in our previous videos. So similar to this podosomes, it also forms a long polymerization actins, F-actins. However, the difference is that it does not have any plaque protein. So this Invadopodia works towards ECM degradations only with this F-actins, polymerized F-actins. And in this case, the MMPs are different. In the podosomes, there are MT1 MMPs. However, in this case, it's just the MMPs, matrix metalloprotease proteins, and the motor proteins is not yet be identified either for DNAs or for MMPs. And it also forms a similar complex. At only thing is the MMPs is different, and also the plaque proteins is not available. And in this case, the MMPs are transferred. The MMPs are transferred with the help of MMPs, or this matrix metalloprotease MT1 are transferred with the help of snare. However, they are not and they are also able to form a complex with this F-actins, DNAs, and with this F-actin and DNAs to make a tertiary complex that contains F-actins, DNAs, and matrix metalloprotease protein MT1. However, this MMP's rules is not presented here in this figure. But this MMPs will also helps in the degradation of the extracellular matrix. Next step is that this motor protein, that is GIP motor protein, along with this RAP8 helps in the transfer of matrix metalloprotease protein, and along with this some other matrix metalloprotease protein with the help of RAP4, and the motor protein is not known, is also transferred. So this will lead to the formation of this further advanced complex for further advanced complex that contains F-actins along with this DNAs, and that contain more polymerized or elongated F-actins along with the DNAs and matrix metalloprotease proteins. And simultaneously as in the case of podosomes, the matrix metalloprotease proteins alone can also function towards ECM degradations. So this is how. And one thing that I need to clarify is that when they are at first stage or at the second stage, the ECM degradation is not enough. However, when they are at this complex stage or this matrix metalloprotease alone, it can degrade the ECM very efficiently. I want to just correct myself what I have said previously. And similarly, in this case, when there is only effectins or these complex formations, there is not much degradations that has been observed. However, when there is this complex formations or only the matrix metalloprotease alone, then there is a degradations that has been observed of ECM. So if you need to understand more about these invadosomes, how it helps in the proteolysis of the ECM, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.